So in previous slides, we already talked about the SEM, and we also talked about the basal lamina. Here you have an schematic that talk about that, and I'm going to try to put these two ideas together. So here you can see that the integrin, yeah, we talk about laminin, we talk about collagen. So we talk that the extracellular need to communicate to the uh, uh, internal cell environment, to the nucleus, and it does it by integrating the extracellular membrane to the SEM and so cytoskeleton and SEM, and how they do it through these important molecules that are the integrins. Yeah, this is how you do it. Okay, so as I said before, I talk about the coherence. Here we're going to start focusing on the integrins, and I already told you that they have different domains. So here you see the transmembrane domain. So here you see again the transmembrane domain of the integrins. Here you see the extracellular space. So this is the extracellular domain of the coherent. Here you see the extracellular domain of the integrins, and here you see the intracellular domain, and here same thing will happen. Okay, and again, those here you can see the interacting with the extracellular membrane with an important protein that we will talk later, that is fibronectin. Okay, so here you find a table that summarizes the different type of attachment, the main function, and the intermembrane features. So this is just for some of you just have to memorize and to remember some of these things. Um, some of these ones were in the previous quiz. Yeah, pretty easy. Now we're going to focus on the cell and extracellular attachment. As I told you, we're going to talk about the focal adhesion points, and we're going to, the main function of this is the adhesion between the cell and the extracellular membrane, and they are the intermembrane features is that they are localized points in the attachment. Okay, so let me show you some examples and I will talk more about this focal adhesion. Okay, so here you find another table. Again, here I summarize the type of attachment. Here you see the space and the associated structure. Here you have more information. You don't have to memorize this, but just so you know, the space here is between 20 to 25 nanometers, so it's very small. Yeah, that is very important. And here the molecules are associated with the actin microfilaments, as we said before. So they will be, the actin microfilament will be it will interact with the nucleus, and this is why you can change gene expression. And this is not the only case of look, coherent junction is also, as we watched in the beginning of the class, is associated with actin microfilaments. And so this is an important, and if you take cellular biology, or if you remember, for example, hemidesmosome is associated with other proteins that are the intermediate filaments, that just to remind you that there is more information about it that you can that I'm not going to go into detail, but I just want to remind you some of those things. So now we're going to talk about the structure of the integrins, and here you see that there are three domains, as I explained already with the coherence. So here you have the first transmembrane domain, then you have here the extracellular domain, and this one will be the cytoplasmic or intracellular domain. Here, in each one of these domain, it has a different function. So in this domain, in the extracellular domain, the idea is that is the integrins is going to attach two components of the extracellular membranes. In this case, it's the fibronectin. This binding site for RGD. RGD means the amino acids that are involved. So, for example, the G means glycine, and the R and GD, R and G, one is arginine, uh, glycine, and aspartate. Yeah, so these are just amino acids. And here, on the other side, in the binding side for the taline, you will find that there will be linker proteins that you will finally attach to the cytoskeleton, yeah, to communicate the signals as we talked before. I want that you see that there is a sulfur here is involved, yeah, as any other proteins, there is interactions there. And here you see one blue section, as we said before, and another purple section. So it's a heterodimer, one that is alpha and beta. Beta that it changed a lot during evolution, and this is why you have different types of um, integrins. Okay, so when we talk about the structures of the integrins, I told you that the integrin contained two large transmembrane polypeptides that are the alpha and the beta, one in blue and the other one in purple in this schematic. I told you that the integrins have different binding sites depending on the subunits. So on here, let me delete all these things. So you have the two binding sites. So on the top, you will see the binding site that attached to the extracellular membrane, and below you will see the one that attach to the linker proteins that later attach to the cytoskeleton inside of the cell. So I already told you that the extracellular part of the alpha and beta subunits will recognize this RGD. RGD are the amino acids, specific amino acids that it will be found in the glycoproteins of the extracellular membrane. So in this slide, we will focus in the interaction between the integrin inside the cytoskeleton. So in this region here, so we already talked about that in this binding site, the integrins will interact with the extracellular matrix. But if you see here, now we're going into the orange region that has a lot of proteins. So here in green you see the cytoskeleton, yeah, in the cytosol. And so the fibronectin doesn't, the integrin, it doesn't attach directly to the actin filaments. There has a lot of linker proteins in between. Yeah, so the integrin tails, I just told you that they interact with the cytosolic proteins that will attach to the cytoskeleton. Yeah, and some of those examples will be taline that you can see here, vinculin, and here you see alpha actin, and they will attach to actin filaments that you see here and here in green. If the question if you need to memorize all these components, no, but you will hear about them in video lecture. So that you know that there is a lot of, again, this is a multiple protein complex. So each one of these, if it's not working, the talent is not working, it will send the wrong signals. So in many of the experiments that you will hear, they are just modifying one of these ones. Okay, so now we talk a lot about different molecules in this class. So you already know these ones that are located here. So here you have the fibronectin that attach to the integrins, that it goes to the membrane, and it has uh, inside of the cell, it will attach to linker proteins that they will attach to the cytoskeleton. Yeah, and uh, all this here, all this will be a focal adhesion. So the fusion, this complex of proteins, yeah, formed by integrins and linker proteins that attach to the cytoskeleton is very important. And again, more names of proteins that they are involved in cancer. So here you have one of the linker proteins, this FAK. You have, we already talked about vinculin, actin, talent. You will hear about this one in the video lectures that is involved again in this sensation, the sensing the mechanical forces. And you will hear these are also important. We're not going to cover them in this course, but this is just also very important. And here you see that the actin and the myosin are always interacting in the movement of the cells. So conclusion, again here, general perspective, you have a, in the matrix, there is a binding site. Here you have later the intracellular signaling. Here you have all the linking proteins that you will attach to this cytoskeleton. And here you have the actin that will send the signals again, as I have been repeating. So all this complex of proteins is called focal adhesion. That is a large macromolecule assembly that they do it through um, that the importance of these multiple complex protein is that they're very important regulating signals. So they turn the mechanical forces into signals that later the nucleus will turn into changes in gene expression. And again, to summarize all these ideas, this is the connection between the cellular environment and the cell. So the integrins are these focal adhesions, and the integrins are in charge of doing this processing of signal. Okay, so now the focal adhesion's main structure and form and function is related to the integrins. And remember that they don't act alone. The focal adhesion is a multi-protein complex, although the integrin is this protein that is linking the extracellular membrane and the actin domain. And this is forming mechanical links between the extracellular membrane and, of course, the cytoskeleton, as we have been talking. Okay, why I talk all this, I give you all this long introduction about focal adhesion and integrins because I wanted that you remember that integrins are involved in a lot of cell signaling. So here you can see the inactive form of the integrin. Here you find the active form with the linking proteins, yeah, that eventually will attach um to the cytoskeleton. And so there is a feedback between the intracellular environment and the extracellular environment. So, for example, the MAP kinase pathway that we talked before, it can produce this type of cluster of cells that it can lead to. So things that are happening in the cell can change the signal that are outside, and in the case it can produce cell proliferation, and the opposite can happen. The signals that are outside can also influence the inside of the cells. And there's future lectures will cover those topics. And I like this schematic that you can see here. Why? Because it shows you three type of signals. So here you see in red, you will see are the oncogenes that we covered in a previous class, the tumor suppressors that are fine in blue in these pathways, and here you just see mediators. In green, you see the outcome of the signaling pathway. So, for example, integrins in this pathway here. So, for example, to a pathway here, it can transfer a signal, and it can be involved in cell movement. So here you have filopodia, laminopodia that are actin-rich protrusions involved in cell movement. Depending on the cell size, on the size of those protrusions, they become either filopodia or lamellopodia. And if you remember, we talked last time about RAC. Okay. Here you have the focal adhesions, and they can be also involved into actin assembly, and it can be involved into anti-apoptotic, migratory, or involved in increasing proliferation. And they do it here. We talked last time about this pathway that is this molecule that is a small GTPase that is Rho. Here they do it through, and I show you that these two are linker proteins attached to the cytoskeleton. Sorry, to the integrins and the cytoskeleton. So here you see how they transduce the signal, and they can produce these changes in the cell behaviors. Uh, Here you see again another pathway that we talked last time, the JNK pathway, that it can have an effect in the apoptosis or the grades of degree of mitosis in a tissue. So remember, there is a dead signal here that it can be controlled by the extracellular membrane, yeah, by the integrins. And here we didn't talk about more pathways that are involved. So here you have the YAP, that is very important. That is another oncogene, and there is an interaction between all these pathways. So it sounds very confusing, but the main idea of all of this is this: that the integrins also interact with intercellular signaling pathways. Yeah, so they can interact from the inside to the outside. So, for example, there are signal like growth factors that can lead to the MAP kinase activation, and they will allow to the clustering of integrins, as I show you here. However, it can go also in the opposite direction, so it go from outside to the inside, and you have different pathways that they can be activated from outside. So because they are sensing the media. Okay, I hope that this all these topics have been clear, and now we are going to move to a class exercise. Okay, so this is the final slide. Here again, in all this lecture, we didn't cover all the adhesions, yeah, all the junctions that are, if you remember, there are multi-complex proteins. I gave you a little bit of information of the adherent junctions that are here apically, because they are very important for the epithelial mesenchymal transition. And here I told you, now we're going to move into the class, into talking about in the course, talking about the what happened underneath the cells. That is the way that the cells invade, and how the influence of the extracellular membrane and the integrins play a critical role in the next step for cancer invasion.